everyone. My name is Jacob Mayer, and today we will be discussing how a virus can serve to eliminate human prostate cancer cells. As I begin, I'd love to provide you some background on prostate cancer to get a better understanding of why this research is so important and why there is a need for new effective treatment options. So throughout the globe, prostate cancer is the second most frequent cancer in men, the fifth leading cause of cancer death, and there are approximately 375,000 deaths per year because of this. While there are various ways to treat prostate cancer, each come with their own limitations and challenges. For example, these cells can develop a resistance to various treatments. Two examples are chemotherapy and hormone deprivation therapy. An example of a chemotherapeutic drug used to treat many different prostate cancer patients is docetaxel. Docetaxel works by inhibiting microtubules from functioning properly, ultimately leading to cell death. Now, this drug is not selectively toxic. So this drug will kill cancer cells, but it will also kill normal cells. And this can lead to various negative side effects for prostate cancer patients. Another form of therapy is hormone deprivation therapy. And this can lead to a more developed form of prostate cancer that's harder to treat, called castration-resistant prostate cancer. So let's discuss some of the key advantages of using oncolytic viral therapy for the treatment of prostate cancer. To start, viruses can be engineered to become selectively replication competent, meaning that we can engineer these viruses to enter into and lyse cancer cells, increase the probability of doing this, while decreasing the probability of these cells doing any harm to normal cells. And the way that we can do this is by using a virus that has a fairly large genome. So we can insert or delete genes in order to maximize the safety and efficacy of the virus. So this therapy can be combined for a combination therapy, with therapies such as chemotherapy, radiotherapy, or surgery. So the overall message is that oncolytic viral therapy is maximizing patient safety by increasing the probability of those cells lysing the cancer cells while decreasing the probability of the virus doing any harm to normal cells. So the virus that we used as an oncolytic agent while conducting our research was equine herpes virus type 1. Now, this is a member of the alpha herpes virus family. Other viruses in this family are the human herpes simplex viruses and the chickenpox viruses. This is an enveloped virus with 12 glycoproteins. It has an icosahedral capsid that encloses the genome, and it has a fairly large genome in terms of viruses of about 150,000 base pairs. It also comes with 76 genes. So why EHV1? Out of all the viruses in the world, why would we want to use this virus? To start, it has a large genome, so that we can insert or delete genes to maximize the safety and efficacy of this virus. Two, we have a good understanding of the life cycle of the virus. And EHV1 has broad tropism. Our group has done research and have been able to show that this virus can infect and lyse various types of cells in tissue culture. And there are antiviral drugs available, such as acyclovir, that can inhibit the virus from completing its life cycle. Now, these are the cells that we used while conducting our research. PC3 is a metastatic prostate cancer with a Gleason score of 4, meaning that it's poorly differentiated and it's a more aggressive version of the prostate cancer. LNCAP and C4-2Bs both are forms of metastatic prostate cancer as well. They do not have a Gleason score. So one of our first research questions, and how we went about finding out the answer to this question. So how much virus is produced in these prostate cancer cells? And the way we went about doing this, so we would infect the cells for various durations of time at an MOI of 0.1. So in our case, this was 100,000 virus particles that we're exposing these cells to. We would then harvest those cells and then conduct plaque assays to determine the titer. So we start with 100,000 virus particles, and then we're finishing with the calculation from this titer. So whatever value we receive from the titer. Now, this is the answer to that question. What we see here are the average of 3 biological replicates of a 24-hour infections across all 3 cell lines. So in the top middle, we see the PC3 cell line, which actually serves as a good model for castration-resistant prostate cancer, which is the type of prostate cancer I mentioned earlier. And what we see here is we are starting with 100,000 virus particles, and we're in a range right now of about 800,000 infectious particles to 2 million infectious particles. So we started at 100,000, we're working all the way up to that 2 million point in T953. And in LNCAP and C4-2b, we see T967 steal the show. We see over 2 million infectious particles in the LNCAP cell line. So we're starting with 100,000 virus particles, and now we're finished with almost 2.5 million infectious particles. And in the C4-2b infection, we work all the way up to over 4 million infectious particles. So we see that large increase in the number of infectious particles in the C4-2b cell line and LNCAP, thanks to T967. And the others performed well and were able to replicate well as well. Now, that was just 2 24-hour infection. Now I'm going to discuss 2 biological replicates of a C4-2b 72-hour infection. So now we're giving the virus more time to complete its life cycle. So what we see here is T967, again, steals the show, working all the way up to 9 million infectious particles. So remember, we're starting with 100,000 still, and we're finishing with 9 million virus particles. And then T953, KYA, and T220, we're able to replicate as well. So in summary, EHV1 can successfully replicate in these tumor cells derived from prostate cancer patients. So we just answered our first question. We now know that this virus can replicate in the cancer cells, and now let's talk about our next question. Our next question is the question of the day. Can this virus eliminate cancer cells? So the way we went about finding out the answer to this question was we would infect the prostate cancer cells for various durations of time. We would then conduct XTT cell viability assays, which would tell us how many living cells are left. Also, I'd like to mention, we're working with 4 different strains of the virus to test to see which is the most effective at serving as an oncolytic vector. So these sets of data right here are the average of 3 biological replicates, like the 24-hour infection of the virus yield assay that you saw earlier. And the solid bar represents 24-hour infections, the horizontal line bar represents 48-hour infections, and the checkered bar represents 72-hour infections. And the theme that we see across all the cell lines is that as the period of time for infection increases, the amount of cell death increases as well. So as you give the virus more time to infect the cells, more of these cells are going to die. So we just answered our next question. We now know that this virus can eliminate cancer cells. So we just answered our questions. We asked, is this virus able to replicate, and is it able to eliminate cancer cells? And now we know the answer to both of them. And we now know that this virus can effectively serve as an oncolytic agent on prostate cancer cells. So the summary of that data. We now know EHV1 can successfully eliminate and replicate and lyse tumor cells derived from prostate cancer patients. And we know as the period of time for infection increases, we see more cell death. And the greatest amount of cell death was mediated at the 72-hour infection. So this right here, these images that you see are infections with the EHV1 LAC-Z reporter virus. And what you're seeing is as the MOI, the multiplicity of infection, increases, you're seeing more dark pigmentation. And what that means is the more dark pigmentation you have, the more infected cells there are. And you see this across all 3 cell lines. So we just answered our questions. we now know this virus can serve as an oncolytic vector towards prostate cancer cells. And now, where does this lead us to? Where can we go from here? We can combine anti-cancer agents to build on previous research done in our lab by a great graduate student by the name of Maria White, and then we can determine if EHV1 uses MHC1 to enter human prostate cancer cells. That is also building on previous research done in our lab, showing that EHV1 uses MHC1 as an entry receptor for cells. And then we would also conduct plaque assays to determine on normal prostate epithelial cells, so RWPE1s. So I would like to acknowledge Dr. Frampton, the principal investigator, the members of Dr. Frampton's lab, Jenny Black, Jacob Kazenelson, and UNCW, the College of Science and Engineering, the Department of Biology and Marine Biology, the NIH, the National Cancer Institute. And as I conclude this talk, I would just like to thank all of you for listening. I would like to thank Dr. Irwin and the other faculty involved in the creation of this event. And I would also hope that you all have a great rest of your Saturday. Thank you.
Questions for Jacob?
Yes.
I was just curious, because I don't know that much about cancer, but when you're thinking of these as therapeutics, how are you thinking of them potentially being delivered in the future? And then in the context, because you brought up metastatic cancer, right? Like, how does, when you're thinking about that, this is a treatment for metastatic cancer, which is more of a systemic, versus more of a targeted?
That's a really great question. So the first part of her question was asking, how do you see this as a treatment for patients in the future? So the way we think about it is, going back to that term, selectively replication competent. So increasing the probability that this virus can kill the cancer cells and decreasing the probability that this virus will do any harm to normal cells. So what we would need to do is we would need to screen for a highly active promoter in the tumor cells. Once we do that, we would need to compare that promoter to the normal cells. And then once we find that out, if there's a difference, if there's a highly active promoter in the tumor cells and this other promoter in the normal cells is not highly active, we'd have to compare the two. We would need to splice out the highly active promoter in the tumor cells, replace that promoter with the immediate early promoter in the virus, and that will lead to a selectively replication competent virus. I actually have a slide to represent what I just spoke on. So the wild-type EHP1 has an IE promoter and IE gene. We would need to find a tumor-specific promoter, insert that into the virus in replacement of the IE promoter. This would make the virus selectively replication competent. Well, the idea would be that it would be selectively replication competent. Does that answer your question?
Yeah,
Okay, thank you. Yes.
Have you thought about other cell lines like breast cancer cells to use the same application?
Okay.
As a virus to overcome?
That's a really great question. Thank you. Thank you. So the question was, have you thought about using this virus on other cell lines? And that has actually already been answered by someone I mentioned earlier, Maria White. She was able to show that this virus, in combination with histone deacetylase inhibitor, valproic acid, is able to have a synergistic killing of a horrendous brain cancer called glioblastoma multiforme cells. So Dr. Frampton and Maria published, I believe it was 2012, and they showed that with the amalgamation of the anti-cancer agent and the virus, there's a synergistic killing, so an increase in the amount of killing of those glioblastoma multiforme cells.
Thank you.
No problem. Are there any other questions?
I just had a quick one. I don't think it's really relevant to anything, but just out of curiosity, were you surprised that the viral yield didn't necessarily correlate with the cell death?
Okay. Thank you for asking. I was thinking about that as well. Well, it could represent that T967 needs more infectious particles to ultimately lyse that cell. So since we saw T967 was in abundance for the most part in the C4-2Bs and LNCAP cells, So while in the cell viability data, we saw relatively similar results. We saw throughout all 3 cell lines in each of the strains, we saw relatively similar results. So basically, I would say I wasn't necessarily surprised, but I thought it was interesting, and thank you for that question, I appreciate it. Yes?
This might be a bit of a random question, but I know that like FDA can get a little selective about even bacteriophage therapy, and I was kind of wondering if you know what the regulations around trying to use a virus for therapy might be like.
Okay, that's a great question, thank you. So there actually is an FDA-approved oncolytic virus called TVEC. Okay, so TVEC is a type of human herpes simplex virus. Thank you for bringing this up because this allows me to connect equine herpes virus to the same family of viruses that TVEC is in. TVEC is a human herpes simplex virus in the same family of viruses as EHV1. So they're both in the alpha herpes virus family. So they have similar characteristics, and that's another reason why we believe this virus can serve as an effective oncolytic vector in the future. So we would need to go through mice model testing, and then, depending on the results from that, to clinical trials. So we would need to make sure that this virus is safe and is able to perform the way we visualize it, envision it to perform. Thank you.
Okay, well, thanks. I think it's time to get to the poster session, so thank you. Thank you to all our speakers for a great session, and we'll see you at the next session.